First of all, Sir Paul Nurse, thank you very much for this opportunity to talking to you about life. What is life? Because I understand you have the answers of this very basic, fundamental, important question, what is life? Looking from the biologist's, yes, perspective. So before going into a little bit of details about the chapter of this book, did you find the answers?
No, I didn't find the answer, I have to admit to you. But I want to tell you what I was trying to do here was to write a book that would identify those things that were likely to be true 30, 50 or 70 years from now. Because many popular books tend to focus quite a lot on things that are a bit beyond the horizon, which often don't turn out to be correct. And the work that I talked about here, which identified principles to think about how we should define life, are ones which I think will still be true 30, 50, 70 years from now, and so will be part of the answer, but they are not the complete answer. And it has to be said, the way I've pulled it together is quite elaborate. It's not a simple dictionary-like definition. It's one which is more generic, more philosophical, if you like, and so may be uncomfortable to some. But I emphasize what has been said here is still likely to be true for quite a long time.
But so the book is What is Life? Understanding Biology in 5 Steps. So these are the steps, or these are the most important steps in order to understand biology, in order to understand life?
Yes, I'm not sure I quite like the 5 steps. I have to tell you that the publisher liked it like that. It sounded a bit like a textbook or a self-help manual to say 5 steps. But it is what they said, and that they should know their business better than me. But it is 5 important ideas, is the way I viewed it. 5 important ideas which are central to life. And if you pull them together in different sorts of ways, I hope illuminate what living things are.
Okay, so obviously you start with the cell, which is the basic unit of life. Yes. And the main, how to say, message, the main thing here is that what is new about the cell?
Well, the message here is that cells have been ignored too much, in my view, compared with, for example, the gene and other parts of biology. And I don't fully understand that, because the cell is the basic unit of all life on the planet. And it's the basic building block, it's the basic structural unit of life, the basic functional unit of life. And as a consequence, it's really critical to thinking about life. And more than that, if you're wanting to know the difference or distinction between something that's living and something that isn't living, then choosing to focus on the simplest entity that for sure is alive, which is a cell, makes it a little simpler to think about the problem. Because as soon as we start thinking about us as a human being, or a fly flying around the room, or a fungus, or a tree, we get tangled up with all the differences between different life forms. But if you focus on the cell, it's actually remarkably similar in all different life forms, and it focuses on what's important. So that's why I think it's a crucial way to start any discussion about what life is.
Okay, just a short stop here. You also, how to say, referred and looked at viruses in this book, which we know that are not live organisms. So then what is their place?
Well, I do discuss this because I discuss it at the end of the book. Mm-hmm. Because viruses are always the life form, let's call it that for the time being, which is confusing in defining life. I come to the conclusion that a virus is alive. And the reason I argue that is that the reason why people don't want to accept that is because viruses can only function inside another living cell. And viruses indeed are not defined by a cell, they're just a structure. Mm-hmm. And therefore, because viruses are completely dependent upon another living thing, it sort of makes people think, well, they're not really quite alive. I made two arguments, really. The first was this: all living things are mostly interdependent upon other living things. So the notion, the idea, the concept of interdependency or dependency upon something else is not critical, because otherwise we'd exclude human beings, because we're dependent upon certain microbes that live within us making certain amino acids. So we're dependent upon them. We're dependent upon the plants that we eat and so on. And I realize it's fundamentally different, but the concept of dependency is almost universal. So that's one thing we should be aware of. And I make the point there in arguing that emphasizes why we should care about other living things, because everything, including ourselves, are dependent upon them. The second thing that I sort of emphasize is that one of the fundamental characteristics of all life is the ability to evolve with natural selection. That is, Charles Darwin is normally associated with that great idea. And viruses do exactly that, as we've seen with COVID-19, evolving all the time. And that is the central cornerstone of definition of life for me. And viruses tick that box. And for that reason, I tend to think of them as alive. And if people don't get convinced by that, and I understand why not, I say, well, another way to think about it is when they are inside a cell and are replicating themselves and making more virus, then we should think of them as being alive. And when they are just a crystal outside the cell, inert and doing nothing, then you can think of them as non-living. And so that is a way out of it as well. I was explaining that to be a little bit jokey about it, but there's some truth in that too.
Jokey or not, but they are very important. Like we've all seen during this pandemic, our life is dependent on these viruses' actions or not. So yeah, they are kind of parasites, even if they are not defined as parasites in the, how to say, medical or scientific term. Yes, actually. Anyway, going back to those 5 important steps in defining, in understanding what life is, you then look at the gene. Yeah. And the chapter is entitled "The Test of Time." Why the test of time?
It's a test of time because it persists in time. Genes persist in time. And this really is something that fascinated Schrödinger, who wrote a book called What is Life in 1944. And he was talking of a code script, which we now understand as the genes, and that they persist from generation to generation, that code script. And it's the test in time, the ability to remain unvarying despite the test of time, which should cause decay, is what is a real feature of genes. And I go on elsewhere in the book to say that the structure of the chemical structure of a gene, which is a double helix, with all that coding information turned inside and protected by the outer molecules on DNA, is the way in which it's stabilized in time. So that's why I use test of time. It survives the test of time, which normally leads to decay, but because it's constantly replicated, constantly repaired, it can remain stable.
Which is a fortune for us, because stability is absolutely, how to say, mandatory in order to be alive.
It absolutely is. If our genes in our body started being unstable, who knows? I might have 6 fingers, or 1 brown eye, 1 blue eye, and whatever. Or maybe my hair would have gone white at age 2 instead of 52 or whatever. Yeah, agree.
And they remain stable in time despite the fact that everything is about evolution. And the 3rd chapter is evolution by natural selection. And the sub-chapter is chance and necessity. And yes, this is the proof that this, how to say, evolution and chance and necessity are all needed ingredients.
Yes, they're great terms. I didn't invent them. That was invented by Jacques Monod, the famous French geneticist in the probably 1970s, something, or 1960s, something of that. But the point about this is, although genes are stable, as I've just been explaining, they do change a little bit. And that's that small changes, the differences in gene structure, that lead to the material upon which natural selection can work. Because you get genetic variation, and that genetic variation is slight. So often the genes still work, but they work in a different sort of way. And that working in different sorts of ways can change how the living thing works. And that can, in some cases, be disadvantageous, but can also be advantageous and result in the ability of that living thing to reproduce more and therefore lead more offspring containing that particular gene, causing a gradual genetic change. And the reason why chance and necessity there is that all arises by chance, but it leads by necessity to improve function. And so these two ideas initially seem counterproductive, because you get design, but you get design by chance rather than design by somebody inventing something. And this is, of course, the big religious debate, because those of a religious view who don't like evolution by natural selection don't like it because it challenges one of the great arguments that was used in favor of a God, which is that we are all too complicated to just have arisen. We must have been designed. But evolution by natural selection provides a way in which that could occur without having a designer.
But this, how to say, by chance, like you've said, might be by biology's chance, not necessarily a, how to say, God's chance. Absolutely.
I don't think it has to involve any, once this is working, any supernatural being. It's biology's chance. What is exactly as you say, it is simply that the mutation, the alterations in the genes are mostly random and they arise by chance. And then selection for reproductive success basically selects out those alterations which are most beneficial to the reproductive capacity and survival capacity of that living thing.
Yeah. And this chapter is the one in which you describe the same, how to say, the two highly related versions of the same gene, one expressed in human, one expressed in yeast. And one of the questions might be, what do we have in common with the yeast?
Well, this is actually my own work, to be perfectly honest. I worked out how the reproduction of cells, of all cells, which I said were common to everything, how the reproduction of cells are controlled, how you divide from 1 to 2. And I worked it out in yeast, which is very simple, described and discovered the gene, the critical core gene that is involved in that, and then asked, well, is there such a gene in humans too? And this was, because it was quite some time ago now, this was before we had the sequence of the human genome, or before we even had the sequence of the yeast genome, which we also did some years later. And so I did a very strange experiment. I took a yeast that was defective in this gene and couldn't divide, and sprinkled on it a gene library from humans. And if a yeast cell that was defective picked up the human gene that could work in the same way, then that yeast could grow and divide, and I could find it, even though it was being controlled by the human gene. And then I could recover the human gene and find out what it was. And doing that, I found that the human gene and the yeast gene were the same, basically, I mean, all extremely similar. And so similar that one could substitute the other. And that meant, since the yeast and the humans diverged evolutionarily 1,500 million years ago, right up here, 1,500 million years, it's 1.5 billion years, it's meant that the way in which all cell reproduction has been controlled has been the same for over 1 billion years. So similar that you can substitute one with the other, which is extraordinary if you think about it.
Yeah. If we think about us humans, how complex we are, indeed. Again, I would stop a little bit here to this cell issue, which was your field of research that eventually led to the Nobel Prize. So what attracted you when you started studying this?
I started it actually when I started thinking about this when I was a PhD student, so age 23 or something like this. And I tried to think what I'd learned doing a PhD is actually doing research is quite tough, difficult job. It's difficult to do high quality, new research, discovering new things. And if you were going to be a researcher, that meant you would fail quite often because they're difficult problems. And if you were going to fail quite often, to keep going, you'd, at the very least, you had to be studying something interesting to justify it, just psychologically. You know? And I thought, what are the important problems in biology? And I thought, well, one thing is we're all made of cells, and all growth and development is controlled by cell division. That's division dividing 1 to 2. If it goes wrong, you get cancer. Major problem. So understanding how cells control their division is an important problem. So I started working on it when I was 24, and I'm now 73, and I'm still working on the same problem. And halfway along, I got a Nobel Prize for what we had discovered, which was the basic molecules that control it. So that's what we discovered. We don't yet know how it all works, and that's what I'm continuing to work on. But that's basically the message. We know the core mechanism as to how it works.
So the message is that you still have to understand a lot of things. It's not done. You are not done.
Do you know, the thing is, it's never done. It's never done. I shall go to the grave saying it's not been done.
Well, in a way, it's, how to say, it's good. This is the, how to say, this is the engine that keeps the world going, no?
It's the engine. You know, we hand over the task to the next generation.
Yeah.
We will have built a little bit of the wall, and they build the wall higher, and that's the legacy.
Do you have the next generation to pass these things? Are young people coming into science?
I train quite a lot of people. I have mostly PhD students actually. I've trained a lot of graduate students and postdocs in my life, maybe 100 of them. And some of them still work on the same area, and they will continue it. And of course, I'm only one of many labs now in the world that are interested in these problems.
But did you manage to attract more after you got the Nobel Prize?
Do you know, I found it, I attracted more people, but not necessarily people who were genuinely interested in the problem, but were interested in working with a Nobel Prize winner. And when I was younger, I wasn't famous, and I attracted people who were interested in the problem. And I think that's probably better. So you're right, I could attract more, but maybe not for the right reason.
Yeah, well, anyway, keep going. Let's go back to the book. So now we reached the chapter number 4, life as chemistry. But life is chemistry, isn't it, actually?
I wanted to emphasize it because it wasn't always thought to be the case. It was now quite a long time ago, it's true, but most of the ideas I talked about were in fact quite old. They're not the ideas of the last 10 years. And life as chemistry really means to say life is, of course, chemistry, but it is only one way of describing it, because life is also information. That's why I used as rather than is, because there's different ways the human mind can look at the problem. And you can think of life in terms of information, you can think of life in terms of chemistry, and in fact, they're interlocked with each other. But the fact that it is chemistry was not obvious to an 18th or even a 19th century scientist. And there was long debates about it because it was thought that life had a vital spark, something that was not explicable in terms of the laws of physics and chemistry. And the point I wanted to emphasize, and my predecessors, of course, did emphasize, is that it is explicable in terms of physics and chemistry. It's extremely complex, physics and chemistry, very elaborate and connected together by this idea of information. But it is basically physics and chemistry. We don't have to look for a new sort of way, a new matter really that would inform it. And you can understand why people thought that there was a living spark. You know, if somebody or an animal or you die, something seems to just, a spark seems to leave them. And that's, you know, all tied up with the soul and so on. And I just wanted to emphasize, no, stick to chemistry and physics. That's a safer way to go.
And information.
And of course, the next chapter was information. Yeah. And there, what we were saying is, yes, it's complex chemistry, but within a very restricted space, we have hundreds, if not thousands, of chemical reactions going on all the time. And the purpose of those chemical reactions are to maintain a cell, to build a cell, to reproduce a cell. And to get that purpose, it has to be coordinated by the flow and management of information. And that coordination leads to apparent purpose. You know, the purpose of growing, purpose of maintaining yourself, the purpose of reproducing yourself. So it's a little strange, but without that information, the whole thing will just be a mess. It couldn't be managed. It would be a little bit like taking a car and disconnecting all the different components so they were all operating independently. It would never go along the road for sure. It would simply be whirring and withering, you know, around and doing nothing. It would have no overall purpose.
Okay, and then before going to the last, it's not necessarily a chapter, it's a kind of conclusion, answering the question, what is life? Yeah. There is a very interesting stop, changing the world.
Yes, yes.
And this refers to?
Well, you know, I wondered whether to put that chapter in or not, because it doesn't address what is life. But what it does is saying understanding life and how it works is important for the world and can change the world. And we have to be able to accommodate as a society, as a human species, really, the knowledge, the scientific discoveries that come from biology, which can sometimes be a bit challenging, and sometimes challenge beliefs that have been held for a long time, and sometimes give us the power to do things that make us uncomfortable. And we need to be able to discuss those things if we're going to use our scientific knowledge for the benefit of humanity and not for the opposite. So I thought a chapter like that would sort of make what otherwise might seem a bit of a remote, almost sort of philosophical thing into something that is real. I mean, that is connected to the real world. So that's why I wrote it, but it is a bit different from the other chapters.
But are we, how to say, using this immense volume of scientific knowledges we have on our own benefit? Do you think that from these pandemics we proved this, that we are able to use, that we trust enough the scientists in order to, how to say, sail safely through such periods?
Well, you know, it could have been worse and it could have been better, you know. We were faced with a viral pandemic, which has been predicted for a century. We weren't prepared for it. That was big weakness in our political system, that we weren't prepared. Even advanced nations in Europe, USA, nobody did very well. we got through, we've muddled through between, you know, all of us. A lot of people have died, but if we'd not done things based on the knowledge that gradually accumulated, many more would have died. So we understood what caused the disease, what the virus was, how to attack it, how to produce vaccines against it, and how to stop the spread. All of this took time. And really, for quite some time, we were very old-fashioned in our approach. We were isolating people. We did that in the Middle Ages with the Black Plague. When a village had the plague, you isolated it. And we were doing the same thing 7, 8 centuries later, because there was nothing else we could do except isolate ourselves, because we weren't even sure how to detect it properly. And so all we could do is isolate communities. Gradually, we got better and better. We now sort of see the end of the tunnel, at least hopefully we do, as long as there's no more variants. And we know that some countries are not well vaccinated, and that's going to be a problem. We're going to have to continue vaccination. So we didn't do very well, we didn't do very bad. But what it does teach us is that we do have to have a better and closer relationship between science and scientific understanding and discovery and political action, both in determining preparation for issues in the future and for implementing them much more effectively. Not just human disease, this was the classic case here, but across the board. I mean, how do we produce alternative energy that isn't producing CO2? How did we know CO2 was a problem? That was a science issue, and many challenged it because they didn't like the challenge it meant to their lifestyles, basically. They won't say it like that, but that's really what it was, that if you didn't believe in climate change, if you did believe in climate change, you'd have to do things that they didn't like. And therefore, rather than do that, they challenged whether it was true or not. And that is a way human beings can react, but they shouldn't react. And our political systems have got to be robust enough to do that. So we had Mr. Trump constantly denying climate change because he didn't like it, not because it was rational. He was irrational. But that is what happens if politicians get separated from knowledge and science more generally.
And still they were comparing with the whole world, still few nations who, or in which the political figures were backed by the scientific figures, by the scientists, standing with them, close to them, sending the message that look, we, the politicians, are taking our decisions based on the scientific decision, results, knowledges. Maybe we still have to learn from this, even if you say in your book, and this here I will quote, the battle with infectious disease is one that we will never win, end of quote. It's a kind of pessimistic or realistic view.
Yes, it is. And one thing I want to say about saying we're following the science is that sometimes we don't know what science to follow, if I can be, because when something like an unknown virus hits us, we actually don't really know what to do. We know there's various things that we need to do to find out what we can then do, so that can be laid out. But for example, you probably in Romania, as well as in the UK, we were being told to constantly wash our hands. Mm-hmm. It is probable it made no difference whatsoever.
But washing hands is good.
Washing hands is good, so it's the reason why it's not been reversed. But actually, it was almost certainly irrelevant to COVID. It's just simply that COVID-like viruses, it did matter, the ones that we knew about before. So it was a good guess, but happened to be wrong. What has been true is we've been very slow to correct that. So everywhere I go, you still have disinfectant and so on to wash your hands, when we know that the important distribution is through air-driven droplets. And so it isn't actually relevant. A sensible scientific proposal adopted across the world that was wrong. What we do know, of course, is isolating yourself did work, as you could imagine, and producing a vaccine was absolutely critical. And that was, so the scientists got it right. But sometimes they, politicians would get up there and say, we are following science. And they hadn't got a clue what science they were following. They just thought they ought to say it. And they need to be more on top of it, for sure.
Yes, but on the other hand, this is how science works. Yes, you make step by step, you discover, sometimes you contradict yourself, not because you lied, but because you discovered something else. So.
Exactly right. So I use the term, I forget if I used it in the book, because I'm thinking of writing another book called What is Science? Science at the level of research, at the front of research, is, I call it, often is tentative knowledge. Mm-hmm. It's tentative, it is knowledge, it is rational, but it may be incorrect. And you will correct it as you study it more. Yeah. And that's an important lesson.
And an important message for the plain public who has no scientific background and who trust the scientists, hopefully, in the end. We will wrap up in a few minutes. Just wanted to point out that this book was written during the pandemic. So from one point of view, this was a very good period for you.
Well, yes, I actually wrote most of it before the pandemic, as I said, but I was finalizing it because I'm not a natural writer.
This is your first book, actually. And this is your first book, and I was actually very surprised.
And I think it reads okay. I'm not being over self-critical, but it needed a lot of rewriting. I would go over the same piece time and time again. And I was just finishing that process off in the first months of the COVID, you're right. So, but I was also actually, it wasn't because I had lots of spare time, because I run an institute here and I was converting it into a COVID testing institution much quicker than the big government initiatives and much closer to the hospitals to protect healthcare places. And so I was very, very busy doing other things.
So this is one experience, a kind of experience that should be, how to say, shared, maybe?
Well, April in the first year, we within 2 or 3 weeks set up, turned our labs, closed the labs down for normal research, turned it into a testing station, and were providing protection for 10 hospitals and 150 elderly care homes. And we were giving them a service of PCR testing with a turnaround time of 12 to 24 hours. And the national system didn't begin to get there for 6 months after that.
Well, congratulations then. One more reason to congratulate you. And what is the Institute doing now? Is still keeping the places, or you just...
No, we stopped testing once their capacity got large, which was, and we then turned it into a vaccination center. But we, all the time, we were still, after the first month or two, we were doing our normal research as well. Because what we did is we provided protection for the Institute as well. So we never closed down, in fact.
Oh, this is, again, an experience that we should maybe sometime talk about when you'll have...
Well, just briefly, I mean, what happened is when I didn't think of it, but when I realized when we were providing testing to protect hospitals, we could protect ourselves as well.
Absolutely.
So we were only closed for about 1 month, maybe 2 months at most. And then we were initially half open, 50% all the time. And then a month or two later, 100%. And we've been like that all the time.
Very, very, very interesting. This is, how to say, true life experience that you should share, again, in the next book, after the book that you were mentioning, What Science Is.
What is Science? Maybe I'll mention it there if I get round to writing it. Yeah.
I wish you to have time to write, to have energy. You have what to write, to put in a book. So thank you very much again for this interview. Congratulations. And I will, how to say, advise the people who are viewing this interview to just read the book, because it's so well written. It has a lot of personal experience there as a scientist. And I understood from this book, also from this book, that you love science.
It's true. I do. And I hope people enjoy it. And I want to emphasize, it's written for anybody who, whatever their level of knowledge of science is. It's hard work in places, but it really is understandable.
It's just, you have to be just curious, and that's enough. You will read this book.
Yes.
Thank you again, Sir Paul Nurse.
Thank you very much.
Thank you. Bye-bye.
Bye-bye.